Welcome everyone to this week's edition of the Extracellular Vesicle Club. And it's a very special edition because it's the first time in 2.5 years that we've actually more than 2.5 years that we've had an in-person and virtual event. So this EVClub actually started some time ago as just something that we would do every month here at Johns Hopkins, and we would have come to a small conference room and talk about EV science. So we are without pizza, unfortunately. We still don't have the pizza back. In the first year of the virtual EVClub, we would make a joke about pizza on the advertisement every week. Joking aside, it would be very nice to have some pizza. But thanks to everyone who has joined us both in person and online from all around the world. We have a talk today also from somebody who gave one of the very first virtual EVClubs. And this is Angela Zivkovic, a very excellent colleague and good friend of mine from UC Davis. She's come out this week to participate in the Extracellular RNA Communication Consortium meeting of the U.S. National Institutes of Health. Today, we're going to be hearing from Angela about some recent work that her group has published on HDL particles in Alzheimer's disease and how they are related to ApoE genotype. She's be emphasizing something that is very important in the extracellular particle field, and that is particle count. How are we doing our counts? What do they mean? Is there any consequence of getting the right or wrong counts? So I'm looking forward to this seminar. And thank you so much, Angela, for agreeing to give it.
Well, thank you so much, Ken, for that wonderful introduction. I'm so happy to be here again. And as Ken mentioned, I'll be speaking with you today about our research on HDL particles with Alzheimer's disease, but I'll also be touching on a couple of additional things that we are really interested in the lab. So first, just to build a little bit of a background, of course, we all know the urgency of discovering treatments for Alzheimer's disease. It's a devastating disease. It's affecting a growing proportion of the population as we have more of an aging population. And one of the things that's really difficult about Alzheimer's disease is that later stages, when the disease is so advanced and fairly irreversible at that point, is about 20 years after the disease process is actually started. So we actually really need to intervene much earlier on before people are symptomatic. But of course, in order to do that, we really need to make sure that the interventions are something that can be safe and effective over long periods of time in healthy individuals before they have any symptoms of the disease. So I would like to make the case to you that cholesterol is a clear and critical intervention target for Alzheimer's disease. So first, we know now it's been very well established that cholesterol accumulation in neuronal neurons leads to increased amyloid beta production. So this has to do with the concentrations of cholesterol has been worked out. This has to do with lipid raft association of the APP protein and the gamma secrete in proximity to each other. Amyloid beta, the toxic amyloid beta peptide is produced. It's also known that cholesterol accumulation in immune cell membranes, likewise through a lipid raft mediated mechanism, leads to increased pro-inflammatory activity. And we also know that activated microglia are associated with Alzheimer's pathology. And then finally, ApoE, which is the strongest Alzheimer's genetic risk predictor, is the protein that it transcribes. It transports cholesterol to and from cells in the central nervous system and is involved in amyloid beta clearance. So we know that cholesterol is important. And just to now link this to HDL particles, so HDL particles regulate both systemic and cellular cholesterol concentrations. So in the periphery, VLDL particles are transporting cholesterol from the liver out to the peripheral tissues to deliver lipids, and then HDL particles bring excess cholesterol back to the liver for excretion. In the CNS, the main HDL particle, which is not an ApoE1 particle, it's an ApoE particle, is in fact transporting both directions, so both from astrocytes to neurons and also taking up excess cholesterol from whatever tissues need of that cholesterol and transporting it back to get rid of the cholesterol or to transport it around. Now, the question is, do HDL particles cross the blood-brain barrier? And nobody has the answer to that. So we don't have an answer to that at this point. Some people say yes, some people say no. But let's, for a minute, just leave that as a black box and actually see if there's any connection between peripheral HDL particles and cognitive function or Alzheimer's outcomes, which there is. So high plasma HDL cholesterol has been associated with reduced risk for Alzheimer's, and that's both in later stages and also in earlier life. It's predictive of protection from getting Alzheimer's later in life. HDL is actually also predictive of longevity in some of the longevity cohorts that have been studied. Low plasma HDL, on the other hand, has been associated with a number of different diseases that are known comorbidities with Alzheimer's disease. And AD patients are known to have lower serum ApoE1. And then finally, and perhaps most convincingly, animal studies have shown the benefits of increasing plasma HDL. So whether we increase plasma HDL by injecting reconstituted HDL particles peripherally, or whether we induce transcription of ApoE1, we see that when HDL particles in the periphery increase, there's an actual reduction of amyloid beta in the brain and an improvement in cognitive function. So again, even though it's a black box exactly how that's happening, it is happening. So great, let's increase HDL. Well, the problem is that we don't really know how to do that. So we can, through drug-based approaches, increase HDL, but we don't know exactly how much is the safe and correct amount to increase it, because it's not a simple relationship like it is with LDL cholesterol, which pretty much the more we reduce LDL cholesterol, the better. And there's doesn't seem to be a lower end point that we can achieve through drugs that is unsafe. Conversely, with HDL particles, there is a U-shaped curve. So you can have too much HDL cholesterol, and this is associated with higher levels of mortality. And again, this is a big question mark. We don't really know why this U-shaped curve is occurring. We just know that it is. So maybe not such a good idea to just indiscriminately increase HDL without knowing what else we're doing. It turns out that it's not just about how much HDL we have, particularly when just measured as HDL cholesterol, but also how functional the particles actually are. So GWAS studies and different ways of looking at the relationship between HDL and various disease outcomes haven't been as straightforward, again, as the LDL story. Low HDL cholesterol can be perfectly healthy. So there are people who have some kinds of mutations that make their HDL low, and yet they're protected from cardiovascular disease. There's also genetic mutations that increase HDL cholesterol, and you can, as I mentioned, raise HDL pharmaceutically, but this doesn't necessarily lead to improved outcomes. And it turns out that HDL cholesterol concentrations really only explain about 40% or a little less than 40% of the variability in cholesterol efflux capacity when you measure that directly. So the amount of HDL isn't necessarily equal to function. And it turns out that there may be as many as 16 different subclasses, functional subclasses of HDL particles that may do very different jobs in the system. So there is growing evidence that HDL function is associated with cardiovascular disease outcomes. So these are just some examples of some large cohort studies that have shown that measuring HDL cholesterol efflux capacity is a better marker of disease outcomes for cardiovascular disease than just measuring HDL cholesterol and even HDL quantity. And so what we wanted to know was whether HDL cholesterol efflux capacity plays a similar protective role in Alzheimer's disease. So there were some pilot studies that indicated that this might be the case. So here's a fairly small pilot study that was published about 10 years ago, where they found that Alzheimer's patients had reduced cholesterol efflux capacity compared to controls. However, the Alzheimer's patients were about 10 years older than the controls, and the ApoE genotype was not taken into account, which is really important because ApoE4, as I mentioned, is the single strongest predictor of risk for Alzheimer's disease. And the population prevalence of ApoE4 is somewhere between 9 and 23%, depending on the ethnic group. But the prevalence of ApoE4 in Alzheimer's patients is 50%. So ApoE4 is overrepresented in the Alzheimer's population. So we did a small pilot study as well, just 5 patients per group here, and also found that controls had higher efflux than patients of both E3, E4, and E3, E3 genotype. So we set out to do a larger study and included 194 individuals from the UC Davis Alzheimer's Disease Research Center biorepository. And we selected controls, MCI patients, so these are patients with mild cognitive impairment, as well as Alzheimer's patients. And all of them were clinically diagnosed within 1 year of the blood draw. We had equal distribution of males and females in all the groups, and it was age matched across all the cohorts. The power that we needed to be able to detect a statistically significant difference in the cholesterol efflux capacity was 20 per group. So you can see we had enough in most. We were just a little bit short in this particular group here, the ApoE3, E4 MCI patients. So HDL are multifunctional particles, and we wanted to make sure to test 2 different aspects of HDL function. There's many more, so this is just the first 2 functional aspects of HDL that we tested. As I mentioned, the cholesterol efflux capacity is really important because it's regulating the amount of cholesterol in the plasma membrane of cells that the HDL particle interacts with. And also because, of course, the protein cargo on HDL particles is really important. For example, the protein alpha-1 antitrypsin lends antiproteolytic activity, and different kinds of HDL particles have different combinations of proteins on them. So we wanted to test cholesterol efflux capacity directly, but we also wanted to look at the activity of the enzyme lecithin cholesterol acyltransferase, or LCAT, because this enzyme is the determining factor in how much an HDL particle can eat from cell verified coagulate in between the phospholipids in a phospholipid monolayer, but you can only fit so much cholesterol in a particle. More cholesterol in a particle, you acid make cholesterol even more hydrophobic, and now it wants to the particle into a that way. So we wanted to also test activity. And so we did that. This is just showing the methods that we used for that. The LCAT assay is a really nice cell-free assay that has beautiful CVs. My student, Brian Hong, spent a long time optimizing both of these assays to make sure that we took down the CVs of the technical replicates so that we could see any change, as subtle as it might be. And then for the cholesterol efflux capacity assay, we use the J774 macrophage cell line. And we normalize the percent CEC that we calculate to an index. So we also always put on every plate multiple control samples so that we can normalize if there's any interplate variability. And so our CV that we've achieved with that assay is about 4%, which is also really huge because when we started, it was about 12%. I'll just point this out right now as kind of like dropping a little crumb for what will be a discussion later, that we do dose the HDL in the CEC assay by micrograms of protein. So that's how we normalize between samples. Okay, so when we looked at the LCAT activity and CEC, when looking at the difference between MCI patients, Alzheimer's patients, and controls, we did not find a difference in the CEC. We found that the MCI patients had lower LCAT activity than controls, but there was no difference in CEC. So remember I mentioned to you this very important protein, this genotype, the ApoE genotype. Well, ApoE4 carriers are at 4-fold higher risk of Alzheimer's disease if they carry 1 copy of ApoE4. And if they have 2 copies of ApoE4, their risk is 12-fold higher than the ApoE3 carrier. So there's lots of different explanations for why that might be the case. People are studying all different aspects of ApoE biology and how that might be. But I will point out that there is a big difference in the binding affinity of ApoE3 versus ApoE4 to both the lipoprotein receptors that this ApoE particle interacts with, and also the lipoproteins that it associates with. So this tiny one single amino acid change in this one critical spot really makes a big difference, such that ApoE4 really prefers to be associated with VLDL particles rather than HDL particles. And it's also cleared more effectively by the LRP1 receptor. And so the outcome is that the lipoprotein profile of different ApoE genotypes is very different. And so ApoE genotype is very influential on all of lipoprotein metabolism. So indeed, when we look at the ApoE3E3 versus ApoE3E4 carriers, regardless of the Alzheimer's diagnosis, we saw that the ApoE3E4 carriers had lower cholesterol efflux capacity and lower LCAT activity. And so now when we look within the genotype, we wanted to see if there was a difference between patients and controls within each genotype. What we found was that indeed in the ApoE3E3 patients, we didn't quite reach statistical significance with the AD, but the MCI had statistically significantly lower efflux capacity than the controls. And of course, the LCAT activity was lower in both patient groups compared to controls. But intriguingly, in the ApoE3E4 patients, we saw this very confusing increase in cholesterol efflux capacity and no effect on the LCAT activity. So we were very surprised, did not expect to see that, and confused, and then started thinking, well, what could be going on here? So why was HDL cholesterol efflux capacity higher in the ApoE3E4 patients versus the controls? Well, we started thinking about, okay, HDL composition is an important factor in determining function. For example, inflammation can really alter and remodel HDL particles. It can change the protein composition on there. The acute phase proteins will displace ApoE1, everything changes. And in fact, there's been some really nice work showing that the more acute phase proteins, in this case, serum amyloid A, that associates with HDL particles, the lower the cholesterol efflux capacity of that particle. So we thought, well, maybe it's composition. But particle size and number are also critical factors. And so I'm just going to take you guys through this exercise here to show you that an equal amount of HDL dosed as protein can give you vastly different results, because you're basically putting different things into the well, depending on what the individual's HDL particle profile is. So just to show kind of an extreme case here, and here in red, we're showing the percentage of HDL protein from small HDL particles, and in green, the percent of HDL protein from large HDL. So in this case, we have someone who has, very few small HDL and mostly large HDL, and I'm just making it deliberately very different just to show the point. And in this case, you have an individual who has most of the HDL protein carried on small particles and very little on the large. So the way it turns out when you put in the right amounts of protein per particle in the two different sizes of HDL, what you find is that in this case, where you have predominantly large HDL, you're going to be dosing with about 20 total HDL particles, 15 of which are the large and 5 of which are the small. But in this case, you're going to be dosing with more than double of that amount of particles. There's going to be 45 small HDL, only 2 of the large particles. And so essentially, if you have a sample that's enriched in small HDL, the equivalent dose of HDL as micrograms of protein, you're actually putting on way more HDL particles on that well. So our question was, well, do the ApoE3E4 patients have smaller HDL particles? Because that would explain this increase in HDL cholesterol efflux capacity. So just to kind of sidetrack just for a second, we do have to really pay attention to how we isolate the HDL particles, because they're kind of tricky that way. There are a lot of them. And since this is an EVClub talk, I'll just kind of give you guys a little bit of a perspective in relation to EVs. There are quadrillions of HDL particles per milliliter of plasma, whereas, there's 10 to the 8 EVs in the same milliliter of plasma. So there are just armies and armies of these little HDL particles everywhere. And they have quite a wide heterogeneity in what they do and in their particle size distribution. And they overlap with things and are close to all the different cutoffs, so they're very difficult to isolate and pull out. So our HDL method basically uses the principle of separating these particles first by density and then by size, because you really have to do both in order to really clear them away from all the other things that might be co-eluting. So we first float up the triglyceride-rich particles in the first spin in our ultracentrifuge. We then centrifuge again and precipitate out all the plasma proteins. And what's left is this kind of middle section that includes HDL, LDL, tons of EVs, not tons of, but the EVs as well. And then we put that onto our size exclusion chromatography system where we separate everything by size. And we then go ahead and grab the HDL fraction with our fraction collector, and we get a really nice yield of concentrated, purified, intact HDL particles. And this is an electron micrograph of them, and it's just this like nice carpet of perfect little HDL that are in the appropriate size range where they're supposed to be. I'll just also mention that because this technique is non-destructive, we don't throw anything away as we're doing this. We're collecting every fraction along the way. And so this method allows us to capture all the other lipoproteins and xRNA carriers like the EVs and the RBPs. So what we also wanted to do was size the HDL particles, and we did so by the gold standard method, which is transmission electron microscopy. You might think it's crazy to try to size by TEM in 194 clinical patient samples, and it kind of is, but I have a dedicated student, Jack Zhang, who decided to take this on as a challenge. He said, yeah, I will try to do that. So what he did was he came up with a way to make the image selection blinded so that it would be unbiased. He wanted to make sure he selected the different images that included potentially weird looking particles. And he imaged more than 20,000 HDL particles per sample. And he developed an automated image analysis protocol to actually be able to determine what's the size of a nice representative sample of those HDL. So here's an example of how his automated image analysis works. There's the image that he captured, then he gets the computer to basically select the HDL particles and to size them. And it just spits out a really nice report of the size of 20,000 HDL particles in each person's sample. So indeed, it turns out that in ApoE3E3 Alzheimer's patients, the particle size is not different from the NCI and the controls. But in the ApoE3E4 group, we do see a lower average particle size in the patients, and that includes the MCI and the AD compared to controls. So what I'd also like to show you here is some of the chromatograms of the actual patient samples that we separated. And what really stuck out to us before we even did all these analyses, what really stuck out to us as we were separating the particles was this kind of missingness of this large HDL peak that we always noticed in our really healthy cohorts. So in the top figure here, you see, this dark blue one here. This is a really healthy patient who has this really nice double peak of large HDL and then also the small HDL. And then you see this is an individual who is young, but less healthy than the other individual. And you can see how they already have a depletion in that large HDL peak. And then here's one of the elderly individuals in red, and you can see how they're kind of missing this large HDL peak. And here are some superimposed images of multiple elderly individuals with of chromatograms of their HDL. And then really interesting here, we had this one morbidly obese patient who essentially didn't have any HDL to speak of. We had never seen anything like this before, and it was a really interesting finding. So the importance of the small and large HDL particles is that they can be functionally different from each other. So the large HDL are structurally different. They have more ApoE1 and other proteins per particle, but they have a lower protein to lipid ratio because they're larger, they carry lots more lipid in the core. And also, certain proteins actually are enriched in large HDL particles and other proteins are enriched in small HDL particles. So they're structurally different, they're compositionally different.
Can I pause for 1 second and ask a question? So between your large and your small, there's clearly two species present. So does that mean these have different biosynthetic histories? Because if it was a small particle maturing into a large, I would expect some kind of a continuum distribution. But here you've got clearly two separate species.
Yeah. So the insightful question is whether there are two distinct species between the small and the large that are coming from two different biosynthetic pathways, because otherwise you would expect just kind of a continuum. The answer to that is actually trickier than one might wish. And I guess the most honest answer is we're not really 100% sure. So yes, there are different metabolic pathways. So first of all, HDL particles are synthesized both by the liver, but also by the intestinal tract. So there's estimates that about 30% of HDL that's circulating is actually coming from the small intestine. And those tend to have a different composition and structure, but not as much is known about them because we don't really know how to pull them out of the whole HDL smorgasbord. And it's also the case that HDL metabolism is a lot more complicated than, for example, LDL metabolism. LDL just gets secreted. They're secreted as VLDL. They have lots of triglyceride. They start to lose their triglyceride, they become LDL particles, and then they eventually are taken up, right? So they just have this unidirectional size-based metabolism. Whereas HDL particles can start out small, acquire cholesterol, be remodeled, be taken up into cells, be spit back out. They can become smaller or larger depending on what cell type they're interacting with, if they're dropping off cholesterol or picking it up. They can be formed de novo from lipolysis that's happening in the bloodstream. So LDL, or I should say VLDL particles are subjected to lipoprotein lipase metabolism, and that generates phospholipid. ApoE1 encounters that phospholipid, boom, you have a new HDL particle. So it's a lot more complicated metabolism. We don't really know why you have these two distinct peaks. There's some really nice research coming out of Sean Davidson's group that perhaps the way that the ApoE1s interact with each other, they form sort of a cage, and that kind of has a deterministic effect on the size of a particle. So there's lots of different possible explanations, but we don't totally know the answer. So, the two conclusions that we come to here is that we probably need different types of HDL for different functions, right? You have all these different proteins that associate with different types of HDL. It must be for a reason. And or that different particle size distributions and compositional profiles are indicative of different underlying metabolism of the HDL particles. So what we really want is precision medicine-based interventions to fix HDL in Alzheimer's patients based on ApoE genotype, because clearly different things are going wrong with the HDL particles in the two different genotypes. And so we need different solutions. But how do we do that? So in the case of the ApoE3E3 patients, we see decreased HDL functional capacity, but no changes in particle size. And in the ApoE3E4 patients, we have decreased HDL particle size, but we don't really see, that there's a difference in functional capacity. So we need different solutions to fix their HDL. So do AD patients need more or less of a certain type of HDL depending on their ApoE genotype? Or is the HDL profile telling us something about underlying aberrant metabolism of the HDL particles? So since the targets are not really clear, we really can't provide any kind of targeted solutions for this. So we're hoping that our new method will be able to help us with that. What we've really spent a lot of time in the lab, and my student Joanne Agus, who is here with us today, has been instrumental in this, is setting up an instrument that is able to measure additional things about the HDL particles as we're isolating them. And we have the expert in the room here, Wyatt Vreeland, who has been very critical for us to be able to set this up and to understand what we're doing with this instrumentation, and who can answer any more technical questions, which is nice to have. And so basically what we're doing is we're adding more eyes as the particles are eluting from our size exclusion chromatography system. So we're not just looking at the UV signal, which is telling us how much protein is coming off, which was great just to guide us to know when to start and stop fractionating to grab our HDL particles. But now we want to know more about those HDL as they're eluting off the column. We want to know what's their size. We want to know what might they have in them. And we might want to know how many there are. And so what we are doing is combining multiple optical detectors, multi-angle light scattering, dynamic light scattering, differential refractive index, and then UV absorbance to try to calculate not just particle size, but also particle concentration. So why is particle concentration important? So one reason is that we can estimate the functional capacity of the HDL on a per particle basis. So for example, CEC per particle. So as I mentioned before, per the same amount of protein, you can have different numbers of HDL particles. So in the case of subject A here, we just have 2 large HDL. In the case of subject B, we have 8 little ones. And so you can see how that can result in differences in the amount of particles that, the amount of efflux that they perform. So if instead we actually dosed by particle count, we would be able to determine whether the higher efflux capacity is due to the quality of the HDL or just simply the number of them. So that can tell us what we need to change. Do we need to change the total number of particles, number of small particles, or their composition? And then in terms of another reason why we want to know particle concentration, because we can do a better job of estimating composition on a per particle basis. So for example, the number of ApoE per particle. So to demonstrate this point, what we've done here is we've measured the concentration of ApoE on small and large HDL. And as you can see, the amount of ApoE is actually higher in small HDL particles. But in this case, in this particular sample, there were more small HDL particles. So when you normalize to particle number, it turns out, oh, actually, the large particles are the ones that are carrying more ApoE per particle, but the small HDL fraction has more ApoE simply because there's more particles there. And we did this in different with peptide. Here you see the amount of amyloid beta. Same, normalized to what you see is that actually the large particles are carrying particle than the small particles are.
Usually these particle counts are from the
particle counts are from our estimated particle counts using the volumetric approach. Yeah. So, when we're simply looking at concentrations in unknown particles number, we could be missing important changes to a subset of the particles. So that's where we want to get to. We want to be able to get to a per particle compositional measure, because that might actually be a much more sensitive diagnostic. So instead of, how much total ApoE is there in the HDL fraction, we can instead say, is there more or less ApoE per small HDL particle or per large particle? So now that we have the way that we can, we hope, we'll see if it works out, that we can assess better what is the composition and function of the HDL particles on a per particle basis and accounting for particle size, can we actually fix it perhaps? So I'm from the Department of Nutrition. I think about food and nutrition. This is what we do. We don't really like drug approaches. We like to do things with diet. So we have done a couple of different experiments to see whether we can modify HDL composition, distribution, and function. And again, the key is because we need to do this safely over many decades. I'm 46. I want to prevent Alzheimer's at 76. I don't want to start taking a drug right now. But if I can do something through nutrition that can help, I'll sign up for that. So we looked in this case at eggs, and we compared the intake of egg whites to egg yolks because we suspected that perhaps egg lipids might be really important for cholesterol efflux. And in fact, we did find that egg lipids increased HDL cholesterol efflux capacity. And what's nice is that actually they did so without raising total cholesterol, LDL cholesterol, or any of the other aspects of like lipid profile. So eggs have gotten a bad rap. It turns out maybe they're actually pretty good for you. And then we did another study where we just for 4 days intervened with a fast food diet of, burgers, fries, and Pop-Tarts for 4 days versus a nice healthy Mediterranean diet for 4 days. And what we found was that the diets completely remodeled the lipidome of the HDL particles in just 4 days. And we also found, I don't show an example of a figure here, but we also found that the glycosylation pattern of the HDL associated proteins was also altered by the dietary intervention. So in just 4 days, we can find out and we can really remodel our HDL. Another approach that we've taken is to actually look at the effects of fasting. So in this case, we performed a study where we fasted individuals for a 36-hour period. So we had individuals come in after an overnight fast. We then had them record everything they ate during the day, and they came in for a 2-hour postprandial blood draw that evening. They then started fasted for an entire 36-hour period. They came in on day 3 at 8 a.m. We took that as their 36-hour fasted time point. And then they ate, again, exactly the same foods that they recorded carefully on that first day. So they fed themselves the exact same foods they ate on the first day. And again, we took a postprandial blood draw at the end of that day. And so what we found was, and this is actually just looking at the cholesterol efflux capacity of the plasma in this case. So we're working on isolating the HDL particles and looking at them. But for now, we just looked at the cholesterol efflux capacity of the plasma itself. And we found that this single 36-hour fast increased cholesterol efflux capacity by 20% versus just a simple overnight fast, and by 50% versus what the efflux capacity is in the postprandial state. So this was really kind of the largest change we had ever seen. And so what was really fascinating was that this intervention attenuated the reduction in cholesterol efflux capacity by the postprandial state into the next feeding period. So even after they had eaten all day, the next day, their efflux capacity was still better than after the postprandial on the first try. The 36-hour fast also decreased oxidative stress and improved inflammatory reactivity of the stimulated macrophages. So it was just kind of all around a really good intervention that had really beneficial effects. What was interesting was that fasting did not, in fact, change either the plasma HDL cholesterol concentrations or the ApoE1 concentrations. So how on earth did cholesterol efflux capacity change so dramatically when neither the ApoE1 nor the HDL cholesterol changed? So that was a big mystery, and we set out to discover why. So here we worked with NMR lipoprofile. We're also doing our own HDL isolations and characterizations as well, but we first sent these samples out for the profile analysis and found that in fact the HDL particle concentration decreased. So there's more efflux with less particles. And it turns out that this is because it was a very specific change. And you'll see that HDL are not as consistent. People have different changes, much more variability interindividually than the metabolic effect of a 36-hour fast. So after 36 hours of fasting, everyone was in ketosis and everyone had higher free fatty acids and all those expected effects. The HDL was also affected, but much more variability. But still what we found was that the amount of the smallest HDL particles decreased and the amount or number of the largest HDL particles increased. And I'll just point out here that, we are talking about these tiny little particles that really aren't that different in size. You're talking about something that's 7 nanometers versus 10 nanometers in size, but that actually makes a really huge difference because a 7 nanometer particle only carries about 12 molecules of cholesterol ester versus a 10 nanometer particle, which carries nearly 10 times the amount of cholesterol ester in that little particle. So this shift in the particle size distribution makes a really big difference in your ability to efflux cholesterol. So we're pretty excited about this potential of precision nutrition approaches to be able to modify HDL particles. And we found that some dietary interventions are affecting HDL particle metabolism, and so changing particle size distribution, like the fasting. Others are altering the relative proportions of different types of particles, the composition of the particles, the function of the particles. And we're excited about it because it's a really nice toolkit of safe intervention that can be applied to individuals decades before they start to show any symptoms. I will also just mention here briefly that, again, because this is mostly an EV-focused group, that we confirmed that our HDL isolation method does in fact also isolate and capture EVs very nicely. So we confirmed that both by electron microscopy, Spectrodyne, and ExoView. And this is all beautiful work done in Ken Witwer's lab here at Johns Hopkins. And then just to mention my conclusions here. So the ApoE genotype is a critical driver of differential relationships between HDL particles and Alzheimer's disease. Using our new system, which is a size exclusion chromatography system that incorporates multiple optical detectors, is looking like is a really nice method for isolating the HDL particles and simultaneously measuring their size and also concentration or count. So that will enable us to do HDL composition and functional analysis on a per particle basis. And this may lead to both better diagnostics and more precise therapeutic targets. This method also allows us to simultaneously isolate the other xRNA carriers that are present in plasma. And again, we're really excited about the precision nutrition approaches that we can develop to fix different problems with HDL. And then finally, some acknowledgement to all of the collaborators and to the funding organizations that have supported all of this work. And I'd be happy to answer any questions.
All right, well, thank you very much, Angela. And thanks to everybody who's online and has probably some insights and questions for us. Let's start with the first question because I think the audience can hear. So the first question was, these profiles that you see that are different between the populations, is it possible that the HDL itself or some component of the HDL could be a biomarker, it could be developed into a biomarker? So first question.
Yeah, that's a great question. I'm, of course, optimistic. However, I do feel it's important to mention that because Alzheimer's disease is, again, it's a disease that takes place over such a long time. And the samples that we're looking at here are all individuals who are already elderly. So the average age, I think, was 78. And so, by the time most Americans get to be 78 years old, they have a lot of stuff going on. And many of the different kinds of diseases that people get by the time they're 78 affect or are affected by HDL, whether it's kidney disease, coronary artery disease, peripheral vascular disease, diabetes, metabolic syndrome, all of these different disorders affect HDL. So as we begin to look for HDL biomarkers for Alzheimer's, it will be very important that it be very specific to Alzheimer's. And it will not be an easy task to do that because so many other diseases that are comorbidities of Alzheimer's also affect or are affected by HDL.
Thank you. And the second part of the question, and we will repeat this, the other question was about the aging population and how much we have, how much data really we have on that process. And so I think we've heard a good response to that. So thank you. I would like to actually ask Wyatt, why don't you come up here?
Okay.
Come up here. I'm going to ask Wyatt Vreeland to come up, and also Joanne, please, could you come up too? Because I have a question that since we are thinking about EVs as well, so not just HDL, but also EVs that will often co-isolate. And I should mention to everybody in the audience, we're going to have a few EV clubs coming up where we talk about lipoproteins and EVs in the next month and a half or so. So I would like to ask the question about these methods. So the volumetric method that's been described today, but then also the EM method. So which of these methods do you think can also be used to understand more about EV heterogeneity? And I'll just step aside so that one or more of you can address this.
So I'll start and then I'll pass it on to you and you as well. So the question is out whether the approach or the approach might be reasonable ways to assess particle size and count. I think that both methods suffer from one main important issue, is separating the particles from the plasma before we're sizing and counting them. And so you have to make sure you're not losing stuff along the way, or at least you have to account for what you've lost along the way. If you have lost a certain percentage, maybe you can correct for that. But if you're losing indiscriminately a certain subset of particles, then it's difficult to account for that. So we're putting in a massive amount of effort to try to make sure that we account for maybe not every of the quadrillions of particles, but as many as possible of the particles that we put into our procedure. And so actually, Joanne has been really focusing on that as the issue. Do you want to talk a little bit about how we're accounting for, making sure that all of our HDL particles actually end up in our HDL fraction and that we're not losing them in other places?
Well, it's really up to method optimization and just making sure that, along the step, as for example, we go through the ultracentrifugation, we have to cancel out all obvious changes such as the speed, the time, are we spinning long enough? And then as we inject it to our HPLC system, how much of along those fractions do we want to inject? Do we want to inject just the top fraction? Do we want to inject the whole entire section? So just really painstakingly going through the purification and steps.
Yes, and this is not, very exciting work. It's a lot of, do I pull 500 microliters from the top of the tube or 750 microliters from the top of the tube? Well, let me see what's in there. So we're being brutally honest about our process and really trying to find all the weak spots and using multiple methods to see what is being left behind. So including NMR, transmission electron microscopy, anything we can get our hands on that has the sensitivity to look at our very, very tiny particles to see if we're leaving anything behind. With EVs, there's, different problems. There's much, much fewer of them. So kind of your biggest issue is not so much that you might be leaving some in other fractions, but are you actually grabbing them and can you actually capture them, because there's much less of them per.
And then I will just
add?
so I'll chime in that we spend a lot of time at NIST trying to figure out how to do particle counts. And particle counting, I'll just say this straight up, is hard. Okay, and it's hard when you've got a really well-behaved sample. So if you compare the particle count, if you take a polystyrene latex standard bead, let's say diameter 10 nanometers, and you try to do a particle count on that with different instrumentation and different techniques, you're happy if you get the right order of magnitude between the different instrumentations and analysis methods. And this is with the world's most well-behaved sample, an isotropic solid sphere of a high refractive index. So scattering shows it very nicely. It doesn't hydrate or dehydrate. It's like the most perfect sample you can imagine. And still, factor 2 is state of the art. Now, if you start throwing in heterogeneity with respect to size and heterogeneity with respect to composition, now you're really in uncharted territories where things are very, very challenging. And so we don't have a good answer for that yet. And it's something we're working very hard on. Basically, with the nanoparticle tracking or particle tracking analysis techniques, you can get whatever number you want by adjusting the different analysis parameters and camera gains and shutters and whatnot. With the TEM, you get artifacts from the fixation or the staining. With the light scattering methods, you'll get artifacts because your particles aren't fully fractionated. So all of the particle counting algorithms that are based on light scattering are assuming that each particle you're measuring or each sample is absolutely monodisperse and uniform, and that's not true. And then if we go to kind of like the one that's probably one of the more promising ones. So we look at resistive pulse sensing. So it's for those of you who don't know that, it's like a Coulter counter on steroids. And so that one is sensitive. Basically, depending on the design of the pore, you're sensitive to different sizes, so you get biases from that. And so then the other one that's promising is cryo. So cryo TEM, where putatively the size and the composition of the material does not change during the sample prep. But then we actually see where we get biases in the particles that show up in the vitrified ice, because when we do the blotting procedure, we get a very thin layer of aqueous that we then vitrify into glassy ice. And because that ice is about 50 nanometers thick, we necessarily get some steric constraints that bias that sample. So the only good solution we come up with something like Honey, I Shrunk the Kids. So you just get small enough you could swim around in the blood and look at each one yourself and have a little clicker and count.
That would be awesome.
But right now we don't know how to do that.
But to just make it a little less pessimistic, in terms of clinical utility, the NMR lipoprofile method is basically using essentially a volumetric approach to derive particle counts in whole plasma. And that's working well enough, clinically speaking. So even though it's not as, correct as we
Yeah, well,
want
I
it,
should
it's working well.
add in, so I am a little bit pessimistic, but that's because I'm a metrologist, and so we're necessarily by nature somewhat pessimistic. So when I'm speaking about these things, I'm talking about an absolute particle count. So this many particles per mil of this size.
Yeah.
But if we're looking at, sample A has 2 times the amount of sample B, that's a ratiometric measurement is much more useful.
Yeah.
And so we're going to find some kind of a baseline where somebody is, not impacted by these deleterious lipoprotein profiles versus somebody is, then that is an easier measurement to do. Ratiometrics are always more easy than absolute. And then, so I have a question for Angela. So the ones, can we go back a couple of slides? It's this one, number 33. Yeah, so here you say, you point out that your HDL particle size, and then you say 7.6 nanometer particles, you get 12 molecules of your cholesterol ester, and then at 10.8, you get 114. So I did a back of the envelope calculation. It looks like that scaling with surface area. Does that make sense?
Yeah. What we're doing is using the volumetric approach, assuming that different types of molecules take up a certain amount of space, and you put in estimates and assumptions that are reasonable about how much space a phospholipid molecule takes up and how much space a cholesterol molecule that's between the phospholipids takes up versus one that's a cholesterol ester molecule that's inside the core, and how much that space takes up.
Okay, so we can talk about it in detail and see.
Yeah, so maybe we can, why don't we go back to, we just have a few questions from the audience, but the first question is from Ursula Sandow, who asks, do the dietary interventions have similar efficacy in young and elderly individuals, or do we know that?
Oh, that's such a great question. No, I don't know the answer to that. I think that there have been some studies, especially with Mediterranean diet, just because that's such a well-studied diet, but there's been some large interventions with Mediterranean diet, and they have seen effects on cholesterol efflux capacity in those cohorts. But I wouldn't say those are necessarily elderly cohorts. So I'm not sure that we actually have the answer to that. We've only ever studied the effects of diet in young, healthy individuals.
Okay.
We'd love to look at the elderly as well.
Well, we a related question. So we have 2 questions left, and one I'm going to go out of order because this one is also about CEC. So given your fasting and diet data and the evidence of plant-based diets correlating with lower dementia rates, do you plan on investigating CEC in a vegetarian or vegan population?
That's a very interesting question. And I didn't have immediate plans to study vegetarian and vegan versus omnivore diets, but I think that's a great idea. Yeah, it should definitely be done.
There, that's another grant.
Yeah,
Trevor,
there we go.
if you want to help out.
Yeah.
And then finally we go to McFarland and we go to Tommy Kobayashi, who asked in relation to AD, do you think that the size of AD affect the blood-brain barrier crossing? So when you go from 0.4 nanometer all the way up to 12, is there going to be a difference?
Yeah, another great question. So there's been some work looking at this in terms of, the porosity of the blood-brain barrier and what size in kilodaltons particles are able to cross with different levels of permeability in mouse models. We, of course, don't have information like that in humans. I think if we had a better understanding of how the HDL particles are crossing the blood-brain barrier, we would have a better answer to that. I don't personally know the answer to that, but I'm not sure that we know really much about how the HDL particles cross. So the question is whether there's any sex-related differences in the Alzheimer's cohort. We did not see any sex-related differences here, maybe because they were all elderly and of course all postmenopausal. So perhaps that, erased some of the difference in lipoprotein metabolism between males and females. But no, we did not see any in this particular cohort. Yeah.
In order to get at the mechanism of dividing the different particles, is there a disease where you see just the large HDL particle distribution? Is that known in the area and no small particles?
There are lots of really interesting lipoprotein metabolism disorders that have been studied. So the nice thing about the lipoprotein field is that it's quite an old field. So, people have been looking into lipoprotein since the 1950s and 60s. And so there are, individuals who have various genetic mutations, for example, in the CETP enzyme that produce a very specific lipoprotein profile. So yes, that is out there, and it would be very interesting to look at those cohorts as well with these new methods.
All right, well, I think we're going to conclude here. So thanks to everybody who joined both in person and around the world virtually. I'd like to thank Angela for speaking today, and also to Joanne and Wyatt Vreeland, Joanne Agus and Wyatt Vreeland, who stepped in and helped to answer some of these questions that I had. Take care, everyone. Thanks for joining today, and we wish you the best with your science and hope to see you again soon.